I'm Rick Prum, and it's a pleasure to welcome you to this lecture sponsored by the Franke program in Science and the Humanities. We have a great EEB turnout. So for most of you, this introduction will be unnecessary. However, this is my first opportunity ever to introduce Gunter, and so I'm going to take full advantage of that. So first, I'd like to thank the Frankes, Rich and Barbara Franke, for their ongoing support for the Franke program. They have so many rich and important connections to Yale today. They're actually hosting the Franke Graduate Fellow Shindig, which is right now over in the graduate school. They are responsible for supporting some 20 or 30 graduate students right now across the university, so they cannot be here. Today's lecture is next in a series inspired by a collaboration with Bob Schulman. Over the summer last year, I said to Bob, hey, we really ought to cook up something related to your new book, and over a series of lunches, we identified basically a theme in the area of philosophy, individuality, and the humanities. So various areas in which philosophy and active engagement with philosophy is important to the quality of science. And Gunter's talk today, in this regard, Gunter is a great example, in-house talent, somebody who's been engaging with philosophy in their work for their whole career. So Gunter is the Allison Richard Professor of Ecology and Evolutionary Biology and a professor in Systems Biology Institute on Yale's West Campus. He received a BS in chemistry at the College for Chemical Engineering in Vienna and a PhD in biology from the University of Vienna. I think that was the first sign that he was exiting the comfortable world of the law-like in favor of the more interesting and rich opportunities to study evolutionary biology. He taught for some years at University of Vienna and then has been here at Yale since 1991. He's been active in the creation of the EEB department in the late 90s. Has served at multiple times as the chair. He's also served as director of the Yale Systems Biology Institute, and he is the editor of two companion journals, the Journal of Experimental Zoology and a special edition of that, the Molecular and Developmental Evolution. Gunter is the recipient of the MacArthur Fellowship and has an amazingly diverse research program. On the side of theory, he's been working for many years on theoretical population genetics, homology and character identity, modularity, evolvability, innovation and novelty, and gene regulatory networks. So these are all fields in which if you go into the late 20th century work and try to dig down and find out what's there, you find Gunter's name throughout essentially the foundations of what are now whole disciplines. On the empirical side, Gunter has been very interested in digit identity and the evolution of the vertebrate hand. More recently, the evolution of the uterus, placentation and the origin of mammalian pregnancy, cell type evolution, and the role of transposable elements in the origin of novelty. Now, I first met Gunter back in 2001 at a meeting in Beijing on dinosaur and bird origins. It was an exciting time for me. I was trying to advertise and get out these crackpot ideas on the origin of feathers, and Gunter was a very sympathetic and interested voice in those early years. It's been a great pleasure to be a colleague of Gunter's here at Yale, and he's really been an inspiration. One of the remarkable things about Gunter and I is that we are intellectual polar opposites. I'm basically a bird watcher, and he's basically a mathematician, by which I mean if you anesthetize us and then brought us back and found out which of our intellectual faculties came through first, for me it would be this natural history organ of the brain, and for him it would be mathematical analysis. Yet despite the fact that we have this utter difference in our sort of intellectual foundations, for some reason over the years we have converged on a major agreement on almost every aspect of evolutionary biology, and I'm still working on species concepts. I think I can get him to move closer to me in that one, but besides that, we're really close, and this has been really fortunate. So we've taught several times in the recent past together, and the experience is the combination of finishing each other's sentences, and then he'll say something that just completely blows my mind in an unexpected way. And so that makes him a fantastic colleague. So today is also an interesting opportunity because we are expanding the usual reception into a book party, because now available in 13 days from Amazon.com is Gunter's new book, Homology, Genes, and Evolutionary Innovation, published by Princeton University Press. So please join us afterwards for an extra special reception celebrating Gunter and the release of his book. So it's a great pleasure to introduce Gunter today, who's be speaking on the metaphysics of homology and the molecular basis of character identity. Thank you, Gunter.
This slide, I know. Yeah. Okay, Rick, thank you very much for this kind introduction. Thank you all to you for coming today and giving some of your time to hear a talk about homology. It's really amazing. I'm honored that you all are here. Thank you very much. It's great. As Rick has already alluded to, at the talk today that we agreed upon a while ago coincides with the publication of my new book, Homology, Genes, and Evolutionary Innovations, and I hope you all will stay and join us at the party. Now, when I started working on this project here to write the book, and actually before that even trying to understand the problem of homology, I quickly realized that I had actually two types of problems at my hand. It's not only a scientific problem, trying to understand a particular pattern of evolutionary diversity and try to tie it to the genetic mechanisms, to developmental mechanisms and evolutionary mechanisms. But at the same time, it turned out to be also a problem that is deeply rooted in metaphysical assumptions that various research programs have. So it turns out that I've realized that the two M's here in the title, that is a strange juxtaposition in the scientific talk, namely metaphysics and molecular mechanisms, actually have to go hand in hand. We cannot address the one without the other because they are just interdependent of each other. And that's the reason why I'm happy to have the opportunity here to talk in the Whitney Humanities Center and the Franke program about this particular topic, because it really spans both mechanistic aspects as well as conceptual and ontological aspects of metaphysics. But let me first start a little bit explaining what homology is about, because even though there is a large contingent of EEB students and faculty here, there are also members of other disciplines here. At the very base of everything, do we have a pointer or anything like that? Laser pointer? Anyway,
someone up here.
There used to be one. That one doesn't seem to have an exit. Anyway, might not be necessary that much. So at the very base of it, the homology concept is a way to think about the basic observation. Does it work?
I don't think so.
Yeah. That even if you compare relatively distantly related organisms, very often you find body parts that you cannot escape noticing that they are in some way the same thing, like tetrapod limbs. Forgive me that I was mixing up forelimb and hind limbs here, but this is besides the point. In frogs, in bats, in cats, salamanders, and even in fossil fishes, we find appendages to the body that, in spite of all the dissimilarities, all the differences in size and number of elements and proportions, give one the feeling that one is still dealing with the same thing in a certain sense. Of course, nowadays, thank you. And how does it work? This week. Okay. In a certain sense, we explain the presence of corresponding body parts as being due to the inheritance of the same thing in a common ancestor. And I chose here 3 examples of a fossil form and 2 recent forms to emphasize that homology is not about similarity. Actually, the hand of a human and the hand of a bird, or the wing of a bird, are quite dissimilar in superficially spoken, the number of elements and how they are fused and so on. And actually the ancestral form not only had 5 digits but actually up to 8. So we talk about the wing and the hand as being the same thing because it's derived from a corresponding part in a common ancestor. Now, homology is one of these concepts that biologists cannot live with it and cannot live without it at the same time, and so you get quotes from the same author actually that on the one hand asserts that homology is fundamental for much of comparative biology and biology in general. But on the other hand, a few years later, David Wake asserts that there's no reason to seek a naturalistic explanation for instances of homology. Biologists should instead turn their attention to questions that can be resolved. So it's a quite pessimistic but quite reasonable assessment, because there is discordance about these topics for about 150 years since this concept was actually defined in pre-evolutionary times. So let me just quickly say why I think homology is so hard, in particular homology of characters is so hard to nail down. On the one hand, body parts, like hands and eyes and so on, are not directly inherited. They are not inherited like DNA. They are not copied directly, but they have to develop in each generation anew. So there's the way how these things are transmitted is quite indirect compared to DNA, for instance. And starting in the 20th century, early in the 20th century, and became absolutely clear with molecular developmental biology, it also became clear that the same body part can actually be dependent on different sets of genes in different species. So the mechanistic underpinnings, at least some of the mechanistic underpinning of the same thing, can actually be different. So we cannot just say, well, the homologue, like the limb, is just an epiphenomenon of the continuity of the genes, because the continuity of the genes is also not happening, right? At least not in a direct way. And then there are also conceptual hurdles that need to be overcome in order to, be meaningfully talking about these issues. So what do I mean by direct and indirect inheritance? As I said, in the case of homologous genes in different species, it's relatively straightforward to understand what's meant by that, because genes are produced by a direct copying process, and actually the 2 copies of an ancestral gene are also, there's material overlap, because the half of the molecule actually comes from the parental DNA. So that is relatively clear how inheritance and continuity of information is possible at the level of DNA sequences. On the other hand, as I mentioned, in anything that is phenotypic, more on the organismal level, everything comes from a single cell, modulo, other forms of rare forms of asexual reproduction, but let's say in vertebrates it always comes from a single cell that and then develops, and there is no direct copying of the multicellular body parts. They all have to go through a single cell stage, and therefore the mode of inheritance is much more indirect than in the case of the. Just one example of many that exist in the literature about the same organ having different developmental mechanisms underneath it is the vulva of Caenorhabditis. And nematodes, all of them have the same vulva. They are clearly homologous. There's continuity of this form, but the inductive signals that are producing the vulva cells in Caenorhabditis and Priscoscus are quite different. And you can make the argument that at least part of the developmental program has actually changed since the most recent common ancestor of these 2 species. So just to make that. And that is not a really new discovery. It was already realized by Hans Spemann and Hilde Mangold. So Spemann was the researcher who discovered embryonic induction, meaning the fact that some cells tell their neighboring cells what to do by some kind of signal. One of the famous examples was the development of the lens in the eye, which seems to depend on a signal from the eye cup during embryogenesis, that if you take away the eye cup, the lens doesn't develop. But it turns out when they did the same experiment in some other species of frogs, they could take out the eye cup and still the lens would develop, which means there's also an alternative ways of inducing the development of the same structure in different species, even within the same sort of larger group of toads and frogs. So that was actually, Spemann was originally trained as a comparative anatomist, and he was very much aware of the importance and the difficulty of the problem of homology. And in 1915 he published a paper on the history and the critique of the concept of homology, in which his conclusion was there is no relationship between any mechanistic understanding of development and the notion of homology in the terms of continuity of the same thing through time. So the question is, of course, very briefly, why is homology as a topic, as a concept, still important? I think there are two main reasons. The one is that homology reflects one of the major patterns of biological diversity, simply the fact that body plans tend to be conserved, and you find again and again the same building blocks in quite distantly related organisms. So in that way, we cannot just ignore it. But maybe even more important for the development of our own science is that I think understanding homology is fundamental for our understanding of evolutionary innovation. I think one of the big open questions in evolutionary biology is to understand how complex organisms arise. And I think we can not understand how novelty arises if we don't understand what sameness means, right? So homology is about the identity of body parts, and innovation is about the origin of such body parts. It is similar to the problem of species origins, only after biologists have understood what species are about, maybe about in sexually reproducing species, the separation of gene pools and so on, was it possible to develop a meaningful and productive research program into the origin of species, right? If you don't know what a species is, it's hard to find out how they originate. And a lot of the research prior to this phase in evolutionary biology, the research was about how to change an organism, but it wasn't about how a new species arises. So that was, I think, in the same sense, we need to have some level of understanding what homology is, what character identity is, in order to understand how new character identities originate, what we need to know in order to understand the origin of a new body part. Okay? And if someone is tempted to argue that nothing is new in evolution, it's actually some people earnestly are doing, I challenge him or her to tell me what corresponds, let's say, to the tusk of an elephant in a Trichoplex species, right? So at some point, these parts that are individualized in vertebrates have to originate, and they were not there before, full stop. There is just no way around it, right? So origin, novelty is a real problem that we need to understand. Okay, that was the introduction. I want to divide the rest of the talk into two parts. The one is talk about a little bit about the anatomy of the homology concept, how it changed over the centuries, and make a proposal that links homology concept with a mechanistic understanding of development. And then the second part, I'm asking, so where is the metaphysics coming in? Why is it necessary to address also philosophical issues, in particular questions of what's real and what's not real, namely the ontology. So let's start with the anatomy of the homology concept. In order to make the communication more efficient, I will first list the main points that I want to make, and then go through them one by one and explain them a little bit more. So one point is one has to realize that homology, as it was originally defined by Owen in the mid-19th century, was about corresponding body parts. It was not about similarities or shapes or whatever. It was about corresponding body parts. Nowadays and after Darwin, we explain homology or character identity as in historical continuity of descent. That is credited to Lancaster, although it's implicit in Darwin. And then my point is that character identity is only meaningful if we talk about individualized body parts. It's not just anything that we can describe or separate out of the body. That it's meaningful to talk about character identity. It has to be developmentally individualized from the rest of the body. And character identity and character states, the different ways how they can be formed, actually I hypothesize, and we'll give you some evidence for that, are based on different genetic substrates. That are certain genes whose job it is to determine character identity, and other genes that determine character states, the way it is shaped in a particular species at a particular time. So the classical definition of homology, going back to Owen 1843, simply says homologue is the same organ in different animals under any variety of form and functions. That is still a pre-evolutionary definition of homology, but has important elements that we shouldn't forget. The first of all, I already mentioned, Owen speaks about the same organ, clearly a body part, not some attribute of an animal. It's also clearly speaking of sameness in under every variety of form and function, and that to me means that he clearly abstracts organ identity or body part identity from the particular character state, the form and function that it takes in a particular form, in a particular species. Of course, the main unsolved conceptual and also mechanistic problem is what we really mean by the same, if it can be of any shape and function, right? So what is then the basis for calling something the same thing, even though it's not even the same thing, right? Then comes Ray Lancaster, who is credited with sort of the first one explicitly linking character identity or homology to descent from a common ancestor. But what I want to point out is that in his definition, he still clearly talks about organs, corresponding organs and body parts. You know, the organ in an animal alpha and organ B in the animal B and so on, as this being derived from the same organ in a common ancestor and so on, right? So that's the sort of the textbook definition that we are using nowadays if we talk about or teach homology in courses. But still, this unresolved problem is here because we still use the term the same character in an undefined way, only that we say it's the same character in a common ancestor, but don't actually say what sameness means here. Now then something bad happened, as usual in the 20th century. Then there was a loss of specificity in the talk about homology. And you can trace this back to the influential definitions by the most influential proponents of the new synthesis, Simpson and Ernst Mayr, where Simpson, for instance, talks about homology as resemblance, whatever, you know, at any level, due to inheritance or common ancestry. Or Mayr talks about attributes of two organisms are homologous, blah, 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 right? And I think this lack of precision leads to a major source of confusion, because in that sense you can use homology for two different kinds of things. You can characterize, say, well, two character states are homologous. They come from the common ancestor, let's say a particular nucleotide and particular position in a DNA sequence, as well as you can use it for character identities, the presence or absence of a body part or the presence or absence of an amino acid in a sequence or a nucleotide. But those are actually two quite different biological realities that we are talking about here, and it's important to realize that using the same term for both notions is actually leading to confusion. Now, I have already used the distinction between character states and character identities extensively, so I probably owe you a little bit more explanation of what I mean by that. The easiest example that I find is if you look at insect wings. Most very good insects have two pairs of wings, forewings and hind wings. In most of the species, it's a wing blade that is good for producing lift. But in some lineages, there is only one lift-producing pair of appendages, like in the Diptera, in the flies, and what are those? Schnaken in English, whatever. Yeah, huh? Yeah, whatever. You have one pair. The second pair, you have a second pair of appendages, but they are not wings in the everyday language, but they are halteres, sort of sensory organs. And in the case of beetles, you have also one pair of wings that are actually used for flying, and another pair, the forewings in that case, is transformed into an elytra, which is a protective cover over the back of the animal. So the point here is that we have actually two character identities, a forewing identity, hindwing identity, which in butterflies can be wing blades. In Diptera, the forewing can be a wing blade and the hindwing is a haltere, or in the beetles, the forewing is an elytra, and the wing blade is the hind wing is a wing blade. So the character identities are forewing and hind wing, and wing blade or haltere or elytra are character states of either forewing or hind wing, right? So that's the distinction. As what I just said, I think that this distinction between character identities and character states, the various realizations of an individualized body part, is important because it also has consequences for the mechanistic understanding. Namely, the idea is that developmental individualization, this is losing its power. If someone else has another laser pointer, that the developmental individuality that's necessary for character identity is actually caused by specific gene regulatory networks that enable the expression of differential sets of effector genes. And I want to justify this conclusion by using actually quite this example of forewing and hindwing identity, because fly development is something that was investigated quite extensively. Now it turns out that for a fly to have a haltere, to have this little club-like appendages on the back of their thorax, requires the expression of a particular gene called UBX, or ultrapythorax. The function of UBX in Drosophila is to suppress a number of genes that are known to be necessary to make a wing blade, to something spread out and be flat and rigid. SAL, wingless, AC, and so on. All of them are inhibited by UBX. So you could be excused to think of that the Drosophila UBX gene is a gene that inhibits genes necessary for making a wing blade. So you could think UBX is an anti-wing blade gene. So it prevents the formation of a wing blade, right? Essentially, right? But if that would be the case, then you would expect that a four-winged species doesn't express UBX in the hind wing, right? But that's exactly what's happening. So here you have a Drosophila with the haltere. This is the embryonic structure that from which the haltere is forming, and the green fluorescence here shows the expression of UBX. In a butterfly, the hind wing also expresses UBX. So UBX is not always suppressing, in all species, suppressing the formation of a wing blade, right? So what is it then doing? It's even present in primarily wingless species in the third segment of the thorax. And that's actually also the case in Tribolium, in Eunonia, which is a butterfly, and so on. But the critical experiment, the really striking one, is a knockdown of UBX function in Tribolium. Okay, so this is the effect of losing UBX activity in the hindwing and in Drosophila and a few other genetic factors, and you get two wings. you could say, well, you just get, you get the four-winged phenotype back. But the same explanation is not possible if you look at the outcome of a knockdown of the UBX gene in tribolium. You get also 4 appendages, but they are 2 pairs of elytra. Okay? So that clearly shows what the role of UBX is not to determine the shape of the second appendage, but actually to determine hindwing identity. And when you take away UBX, you are left with 2 identical appendages that are both of 4-wing identity. Okay? So UBX is not determining the shape of the appendage or the phenotype per se, but it is one that determines that the second pair of appendages is a hindwing identity rather than forewing. And if you take it away, you get forewing identity as a default state. Okay? So that already shows that UBX is a character identity gene, not a gene that determines a particular phenotype. So character identity and character states are 2 aspects of body organization that are the result of manifestations of different developmental mechanisms. The problem, though, is that if you manipulate a gene that affects character identity, you also get a different phenotype, right? But you can, in principle, and in evolution it happens, have different character states with the same character identity, with the same character identity genes being active. Now that led me to this cartoon model that sort of summarizes how character identity and character development, or body part and even cell type identity is working. And I posited there are sort of three levels to be distinguished. The one is positional information signals, the inductive signals. The one cell tells the other cell what to do and where it is and what time it is. And that interesting thing is, so these are the things that we have seen in nematode development that are quite variable. That is the examples of inductive differences in eye development that Spemann and Mangold have discovered. So positional information, even though absolutely necessary in the development of a particular species, can be variable between species. Okay, that is all the examples where developmental mechanisms seem to be different for homologous genes that I know of can be interpreted as difference in positional information signaling. The positional information signals then activate what I call a character identity network, or gene, that also can be called core regulatory network in cell type identity. And this level of regulation is actually quite conserved. I will give you some examples later. And this network mostly consists of transcription factors and transcriptional co-regulation cofactors, as well as probably also long non-coding RNAs, and are quite conserved and quite rigidly associated with character identity. And they then regulate what we could call realizer genes, the genes that have gene products that actually do physiological work, like enzymes, contractile cytoskeletal proteins, extracellular proteins, and all of that, right? Now the important point here is if character identity is actually associated with character identity networks, it's quite obvious that the same character identity network can regulate different sets of realizer genes, because it only depends on the presence or absence of particular cis-regulatory elements in the realizer gene, whether they are either suppressed or activated by it. Now that explains how mechanistically character states, the phenotype, can be dissociated from character identity, because the same character identity that allows differential regulation can actually differentially regulate different sets of target genes, right? So that's so my answer to what does it mean to be the same thing under every variety of form and function. Because form and function depends on the realizer genes that you are expressing, while character identity fundamentally is the ability to be different. How different or in what way different is open, right? So that's what I'm saying here. Character identity networks enables the expression of different realizer genes, but do not rigidly determine which realizer genes are expressed. That can change in evolution. So different sets of realizer genes can be controlled by the same gene in different species. So that sort of allows for this strange conceptual dissociation between being the same thing under every variety of form and function. So what the gene, thereby the character identity network, actually does is enables the expression of different realizers genes. But in a way, for our philosophically trained colleagues here, one could say character identity is the condition for the possibility of being different. So it's the Bedingung, Bedingung für die Möglichkeit von, a transcendental figure of thinking, like in Kant. It is not a particular outcome, it is the possibility for particular outcomes, right? Just so that I assert that core regulatory networks is not just something that I made up on the weekends. There's actually quite a number of cell type identity networks worked out. Here is one example for liver cells, and all of these character identity networks always have the character of actually positively regulating each other. So they are all necessary for each other and stabilize their expression relative to each other. Another example is the core regulatory network for embryonic stem cells with 3 members. And in this context, I also want to point out that another characteristic that is in particular important for the lab and the work in my lab is that the transcription factors that are coded for by the genes in the core regulatory networks are actually obligatory, interacting, and cooperative to affect the expression of any target gene. So they are not just a combination of independent transcription factors, but they actually have to cooperate. And I think that's also the reason why they are evolutionarily conserved, because they consist of genes that code for co-adapted proteins. They cannot just replace them with another transcription factor of the same kind. Another example is the highly conserved gene regulatory network underlying the development of insect eyes. It is also known to be extremely conserved in contrast to the inductive signals. So these character identity networks are genes, mostly transcription factors and cofactors, that are jointly necessary for the character identity. And the transcription factor proteins coded for by these genes cooperate in regulating either each other as well as the downstream target genes, and they are autoregulatory, have a positive feedback. Okay, so that's the basic idea. So how does this work out and help us develop a research program to understand the origin of novel things, novel identities? If this is correct, then evolutionary novelty is a novel character identity. And if we want to understand the origin of a novel character identity, we have to understand how the character identity network for the new body part originated. Now, with this hypothesis, which I say it's a hypothesis, we don't know whether it's true or generalizable, but at least with this hypothesis, we have something specific to test experimentally, right? The best example that I know of that comes close to this notion is the work by Artyom Kopp from UC, not Irvine, Davis, on the evolution of a special character on the male forearms. They call the sex combs that they use for copulation. It's a novel character within a subgroup of Drosophilids, and without me actually influencing what he published in 2011, he actually also came up with this model that the origin of the sex combs arises through the derived feedback interaction between double sex and sex combs reduced. Sex combs reduced is necessary for transverse bristle row determination, and the interaction with double sex is the one that then forms these individualized bristle rows that are known as sex combs. In our own lab, I'll just very briefly say how we are going about using these ideas. We are looking into the evolution of a novel cell type, in particular the sidual cells that are part of the uterine endometrium that are necessary to deal with the invasive placentation. Koryukin has shown that there are cells, endometrial fibroblasts in marsupials, in the opossum, but no decidual cells, and we are trying to understand how this is working. Our current model is that a particular regulatory gene, FOXO1, was probably recruited into the regulation of these precursor cells, and so we are currently working both on the cis-regulatory changes that lead to this recruitment, as well as we have already published extensively on the evolution of transcription factor cooperativity between some of these genes and the novel derived genes. So just to tell you that it's not just talk, there's real work behind it as well. Okay, now a little detour into philosophy. What does this have to do with metaphysics, and in what way is talking and thinking about homology has to be linked to talking about metaphysics. Even in the book I have a chapter in there that's called The Long Shadow of Metaphysics on Research Programs, and I fully expect to get a lot of flack for that, to have a metaphysics chapter in a science book. But the reason is actually expressed here in this epigraph from Michael Gieslin, who wrote, it should be clear by now that metaphysical preconceptions profoundly influence the course of scientific investigation. I think that's, even though we don't reflect on metaphysical issues in our daily research, we make implicit assumptions about metaphysical concepts that sort of are the background and also the limits for what our research programs actually can achieve. And I think that's, metaphysics here, I mean in particular the fact that each science, each mature science has its own ontology. And our own ontology meaning just a list of things that we think is profound, are real, that is necessary to talk about, and the other things are not, not of our concern. The easiest for me is the ontology of chemistry, at least sort of in a superficial way. What we talk about in chemistry is atoms and their parts, neutrons, protons, electrons, molecules that are, atoms stick together, ions that, atoms that lost or gained some electrons, and complexes that are molecules that are hanging together by weaker forces. But essentially that's what we're talking about, right? And that's everything that's strictly chemical of nature is in one way or the other can be expressed in terms of atoms, molecules, complexes, ions, and so on, right? Now the same is true for evolutionary biology, the new synthesis biology, that has a very clear ontology that things that we are willing to talk about in this paradigm. There are genes, in particular different alleles, haplotypes or genotypes, phenotypes, but only sort of as a vehicle for other genes. Then we talk about populations, but mostly populations in the sense of gene pools as collections and frequencies of genes. And then species, which are essentially separate gene pools. So that's roughly what a population geneticist is allowed to think about, right? And then there's some concepts that go with it, linkage, disequilibrium, additive effects, and so on. So the point that I'm making is that there's a hole in this list, in this ontology, and that is the black box where the organism should be, right? The individual, the organism that actually does biology, right? That reproduces, that does things, that eats and defecates and everything, right? Mates. And so, but that is not part of that ontology. We have no explicit way to talk about them, right? About their structure and their importance at all. They are sort of somewhat folded into the notion of fitness, which is the only phenotypic character that a population geneticist really cares about. And that is a reflection of what has been called the hardening of the new synthesis. I think it was Stephen Jay Gould who came up with this term, said that that leads to a notion of evolution as just changes in gene frequencies. That's it, right? But if you only think as evolution as changes of gene frequencies, it's very hard to introduce any notions of phenotype complexity, of novelty, of different forms of life. Not even plants and animals exist in this sort of reduced language, right? So in talking about homology, about innovation, you cannot do it within this ontology. So you have to find out what kind of concepts or categories we can use in order to talk about. Now, one insidious consequence of this way of thinking is that if natural selection is the only causal factor that you are accepting as an explanatory model, then any explanation has to remain within the limits of a species, right? Because natural selection is only happening through competition among members of the same gene pool, and that's the end of it. So meaning that when a population is splitting, what's going on in this population has nothing to do with what's going on in the other population. So any causal narrative has to remain within the limits of a single species, right? On the other hand, any type concepts, homology concepts, are always trans-species, trans-specific comparisons. And that really leads to a problem, is this at all meaningful, right? And thankfully, two colleagues have expressed this quite clearly. Reeve and Sherman took the pains in 1993 to explain that any talk about phylogenies and comparisons in homology is nonsense, because it would mean that the ancestral species would mysteriously reach from the past to clutch the throats of the descendants. Meaning that any talk about things that go beyond the limits of species would imply that there's some kind of causal connection that cannot exist, because causality ends at the boundaries of species, right? So that shows how difficult it is to overcome or to extend our thinking if we stick to the existing ontology of a particular research program. So we can ask about alternative ontologies. One is then say that homology is an illusion. We only a residuum of similarity due to common ancestry, and it's not something that we need to be concerned about, right? That's certainly a possible position, and prominent biologists and philosophers have defended it. But there are some alternatives. The other alternative that we have in the classical philosophical literature is to talk about natural kinds and essentialist natural kinds, meaning that a thing is a particular kind of thing if it has a particular essence. Like, for instance, to be a carbon atom, that you have to have 6 protons. That's the essence, right? And so it has to have some intrinsic property that makes it what it is, right? The argument, however, is that in evolutionary biology in particular, then an argument by Michael Gieslin and many others, is that in evolution there is no such a unchangeable essence, and therefore they like to talk about individuals, that are things that have no essence, that have no intrinsic necessary properties. They have a definite beginning and a potential end. So they're historical entities. And they don't have instances, independent instances, but they only have members. Like the members of the same species are part of this process of the continuation of the species, reproduction, and so on. While the carbon, you have different instances of carbon atoms. They don't need to be part of one functional system to be still a carbon atom, right? So those are the two ontological categories that we have as at present, namely either essentialist natural kinds or individuals. Now the problem that I have with it is homology, the way I think about it, fits neither of those categories. Homologs, I agree with Gieslin, have a definite historic beginning as novelties and potentially an end. They can die out if all their lineages die out. So in that respect, they behave like an individual. They form lineages of descent. So in that sense, they are like you can draw trees of descent and modification of a particular organ. But they have some essential properties, namely they need to have a mechanism that makes them different from the rest of the body, right? They need character identity network In that respect, they are not like individuals like Gieselin has thought about it. And they're often conserved with respect to some organizational features, you know, your limbs and brains and things like that. So I think we need a new ontological category, which I could call homeostatic individuals. They have a beginning and a potential end. They form lineages and have historical continuity, but they require specific mechanisms for the individuation. In that respect, they have some things of like an essence, but not an essential, you know, shape or form or function. And the individuating mechanisms, if my model is correct, are actually homeostatically maintained because of the necessity of protein-protein interactions for their transcription factors to do their work, right? So they are homeostatic, like homeostatic property clusters of Boyd. They are still lineages, historical entities, and they have some things that look like a little bit of an entity. And I think I now used up my time a lot, and I thank you for your attendance and your attention. Thank you very much.
Great. I'm going to let you use your or take your own questions, but just to help with the recording, I'll ask that when you ask a question, you use this non-microphone or this microphone that's only being recorded on the tape. So questions, and you can take them, and I'll hand out this mic.
Gunter, so how do you see identifying character identity networks as kind of the way of identifying evolutionary innovations in changing our approach of thinking about evolutionary innovation as from the empirical perspective? How will we actually identify and individualize the sense of hypothesis of evolutionary innovation?
So I want to emphasize that not for every homology hypothesis that you propose in a systematic study, you need to identify the character identity network. That is also not the case by describing species. You have to, in every instance, actually experimentally show that they cannot make babies with the neighbor species. There are lots of indirect criteria that you can use to make it plausible that one species is distinct from another one, you know, population, genetic, geographic, and other evidence. And I think the general, you know, morphological criteria to identify homologs work actually pretty well. I think it only becomes necessary to refer to a model or this model or some other model if you want to understand mechanistically what happened when in one descendant lineage something new appears that doesn't seem to have anything corresponding in a sister lineage. Then I think you have to do the hard work and actually try to find which part of the developmental mechanisms are responsible for character identity and which ones are not incidental, and then try to understand how recruitment of genes or adaptation of transcription factor genes leads to a new coherent character identity network that then gives rise to a new individual that has evolutionary change of its own, an evolutionary history of its own, different from the rest of the body. Thank you. Jacques? Sorry, Susan. Yeah, no, please, yes.
So I was thinking about the last thing you said and your sort of new definition of the individuals as homeostatic, and I wondered if you had applied that concept also to these ideas of evolutionary transitions. So we move from molecules to cells and cells to multicellular and on up. And if you would apply this to any of those levels of organization, or really just individuals as we think of as an organism?
No, it's so that the idea is that at least complex organisms consist of individualized parts. And so in individuality, I mean any parts that is of a multicellular, mostly multicellular organisms that acquired developmental and genetic individuality that makes them able to, to react to selective pressures differently from the rest of the body, right? And thereby, take on a specific function and so on. So individuality is in this more general sense that starts at the at the cell level all the way up to maybe even the zooids of a colony, right, potentially. I don't think it's useful and actually necessary for any subcellular things, I mean, at least with molecular entities like genes and so on, because there's something else going on there, direct copying and indels and recombination and so on, that I think is mechanistically a different story than what's going on with phenotypic body characters. Jacques?
Hi, Gunter. Great. So, where does the gene itself sit in this hierarchy? Where are you on the side of, should the homology relation be at the gene or its sequence? Or the
sequence determines the character state or the allelic identity of a gene, right? It's technically sometimes not easy, but in general, a gene is the corresponding DNA segment that has been passed on from common ancestors on and on, right? Of course, the sequence itself,
but maintain its function, right?
Right,
So
its function and its identity is something that gets duplicated and only in one copy transmitted to the next generation. I mean, there are special cases like the ribosomal genes that have all this homogenization, where actually it's not the individual gene that's the historical entity that changes over time, but it is the whole family of ribosomal genes, because they homogenize each other, so they remain more similar to each other than any instance in another species. So it's a little bit more complicated than that, but one always has to ask at what level is historical continuity arising, and what maintains it. I think in the case of these ribosomal genes, there's different mechanisms, there's gene conversion and duplication and loss of genes that homogenize them and make them one unit of evolutionary change, as opposed to other single genes. It's so I'm thinking that there is no simple answer that cuts in all directions, but we always have to ask where, at what level of organization historical continuity arises and what causes it.
let's take this up to another level then too, with about cultures that reproduce themselves and memes, and how does that notion fit in
defer
there?
to my colleagues from the Humanities to answer that question. I shouldn't answer that, but of course, as a Yale professor, I cannot restrain myself to answer it. Cultures are a historical entity, right? They exist because they are transmitted, at least elements of it, transmitted from generation to generation. And you can make an argument, whether it holds any water, that is not me to answer, that some elements of those cultures have individual continuity, right? Organizational institutions, like we discussed last night. Institutions have a life on their own. They have a historical continuity. They are independent to some degree of the people who represent the institution, because individuals come and go, but they have a rule system that makes sure that the institution remains the same thing. It remains a university, or it remains a church, or it remains a military organization, right? But each of them have this property too. Whether any of those ideas help us understand the identity of those institutional identities, individualities, I don't know. That needs to be answered by people who really understand their work.
So I'm interested in character states and character networks, and if you take something as simple as a virus, are any changes in a virus ever going to meet your definition for novelty, or are they all just going to be changes in character states?
Well, I mean, if you duplicate a gene and it differentiates, right, and creates, let's say, a new code protein that then is inherited, and each of these two copies have, take on independent functions, then you have a novelty at that level. I don't think any of the ideas that I talked about networks and so on, might be useful there, because mostly what I thought about is a consequence of the fact that these structures are made up of multiple cells. Whether there's something similar going on at the gene regulatory level in viruses, I don't know. This is something you have to tell me at the next committee meeting.
Only a quick comment in regard to the gentleman who raised the issue of culture. I think that this is extraordinarily rich and extraordinarily powerful, because I think that at another level up in biocultural coevolution, there are in fact moments at which there are emergences of complex networks of systems that did not exist before, that then come to be channeling mechanisms whereby
Right.
a reproduction of the kind that you're calling homologs will in fact persist across cultural histories, and not only hominin cultural histories, but cultural histories in other species as well. So I think it's fantastically interesting work. Thank you.
Okay,
That's all.
Thank you. Very kind of you.
Other questions? Well, let's finish there.
Okay.
No, we have one more? Yeah? Great. Well, please join me for reception right directly downstairs, and let's thank Gunter again.
Okay, thank you very much.